I'm Jacob from GenScript. And me and my colleague Fengmei is going to give you a presentation regarding how we accelerate the innovation of nucleic acid. The title is Harnessing RNA to Simplify and Advance Gene Therapy. So CRISPR has become an increasing acceptance in various fields of biotechnology like gene therapy, like treating infectious disease. Casgevy, a recently approved, FDA-approved drug, the approval of Casgevy showed the great potential presented in those non-viral-based gene editing technology. So here comes the first part. It's the ultra-long guide RNA for the second-generation CRISPR toolbox. I would love to show you guys how we are able to mitigating those challenges in ultra-long guide RNA. So with the approval of Casgevy, so FDA has keep posing the concerns about the potential off-target effect created by a Cas9 system. So after some literature screening, we believe that by applying these ultra-long guides like pegRNA, ATGRNA, and ePegRNA could dramatically lower the off-target effect, which may do some good in the applying this technology in gene and cell therapy area. However, when we look at the pegRNA used, usually is 140, 170, and 190, which is pretty long. So we believe that there are some potential challenges for synthesizing those long guide RNAs. As I just mentioned before, with the opportunity presented for prime editing, there are a big challenge for those technology. One of them is the challenge of synthesizing long guide RNA. As I just mentioned, the length was above 190 nucleotides, some of them even above 200 nucleotides. And based on the iterative reaction feature for oligonucleotide synthesis, it would be very crucial to control the reaction rate for each cycle, conversion rate for each cycle. Without a good conversion rate, we are barely able to get to the end of the synthesis. And today we're going to show you how we can mitigate those challenges. So we believe there are 4 key aspects to the success of long guide RNA. So first is the solid support, then is impurity control from starting material, and the third one is the production process. Last but not least is the purification process. So to start with, solid support is very key to the synthesis because the oligonucleotides grow on the solid support. So there are multiple types of solid support presented, including those polystyrene beads, which the advantage of that is the high loading amount, which makes it quite efficient to produce a mass amount of oligos. However, the drawback is the length was limited to below 30 nucleotides. So it would be pretty good for synthesizing oligonucleotide drug like ASO and sRNA, and it won't be efficient to synthesizing even sgRNA. Aside from polystyrene support, we also have CPG support with various pore size. So the advantage of increasing the pore size is it allows us to synthesizing longer oligos when the pore size increased. However, the trade-off is the corresponding loading dramatically drops with the increasing of the pore size. So there's a balance. So especially when we are trying to synthesizing those guide RNAs, sgRNA, 100 nucleotides, maybe the CpG 2000 angstrom would be good, but when pushing it through or pushing over 150 nucleotides, we have to design or find our own support. So GenScript has leveraged its potential to figure out that we can find a solid support with moderate loading, but a good capability for synthesizing longer guides, especially over 150 nucleotides long. So here is the graph for the crude product, so you could better illustrate my point here. So when using a random vendor, you can see when synthesizing a 173 nucleotide guide RNA, there was some of the full-length product, but the majority of the peak is the early elute impurity, which normally stands for N-, and there was some late elute impurity as well. While switching the solid support to our GenScript support, you see dramatic increase of the full-length product and decrease of the early elute and late elute. We believe that by applying the correct solid support, we are able to first achieve the maximum synthetic efficiency of the long guide RNA. Aside from the solid support, impurity control is one of the things that has been mentioned by multiple vendors or even the amidite suppliers. We have listed that there are multiple impurities presented in the phosphoramidite. And certain of them have some drawbacks, like for this one, it would have an N plus 53,000 impurity, and this would lead to N plus, this would lead to N minus. So here we would love to show you a case study of how we study those impurity and the effect, or evaluate the effect of that impurity. So let's take this one as an example. So these are C stands for the normal phosphoamidite, but without the protecting group on the C. So we have actually deliberately mixing those RC into the normal amidite to see the corresponding reaction efficiency. So what we figure out that is while we were lowering the amount to 0.2%, then there is a minimum effect presented in the synthesis. By increasing the amount, we would see a dramatic increase of the early elute, which stands for the N- impurities. And here, when we know the ratio, then we go ahead and developed our analytical method to verify the presence of the RC impurity. And by this linear regression, we can tell that the LOD value could be reached to as low as 0.05%. So this is much lower than the alarming limit. So that allows us to control the presence or the presence of the impurity. And by that way, we'll be able to mitigate the potential drawback generated by those impurities. Aside from the impurity control, production process has a great effect on the success of the long guide RNAs. So GenScript has multiple synthesizers, including this high throughput synthesizer, the 192 scale synthesizer, where you can synthesize in 192 sequences at the same time. And we also have the scale up instrument where you can go with one sequence at minimum scale. So we envision that those scale-out strategy would be pretty efficient while people are doing some early stage screening. You need a bunch of different sequences to be synthesized at the same time and figure out which one works the best. While you figure out the PCC sequence, we also have the corresponding scale-up strategy to allow you to get to the toxicity batch or even the IND or clinical trial 1, 2, 3. So last but not least, purification is also the key factor. What we normally know is since the relatively low coupling efficiency of the ultra-long, when it comes to the ultra-long guides, so we have to apply HPLC purification technology. In that way, without that way, we are unable to get the final product. However, HPLC has its drawbacks. First of all, it's gonna take a little bit longer because you need to run it through the HPLC and then the centrifuge or lyophilize down. So the turnaround time will be a little bit longer, but that is not the biggest issue. Normally, by applying HPLC, the cost would be relatively high. So this is not a very cost-effective approach. So we believe that by doing all the controlling strategy we presented early, we are able to provide a desalt grade ultra-long guide to the community to test out, get a understanding of how that goes. So the advantage of the desalting is that the purity was moderate, the efficiency would be moderate, but as a trade-off, the turnaround time and the cost would be dramatically turned down. So that would make this approach quite cost-effective during the screening stage. And this graph on the right gives a great illustration of our point here. So this one is an unoptimized desalt strategy for 173 nucleotides guide RNA. There are some early elute impurity and late elute impurity presented there. But by applying an optimized desalting strategy, we are able to lower the ratio of early elute impurity and late elute purity. So this looks like the moderate one. So we believe this would allow the community to test that out in a cost-effective way. So aside from the PD work, we also have adopted or developed a multidimensional analytical method to understand the quality of the long guides in a comprehensive way. So we have analytical method, including the physical-chemical method, bioinformatics method, and even cellular-based assays to evaluate the quality of our long guides. So this is the normal UPLC method. You can see that, so here we put an example of 118 nucleotides long guide RNA. So the purity for HPLC purified guide RNA was relatively good, while the purity for desalt grade RNA, yeah, you see some early elute impurity but late elute purity. The purity was actually quite moderate for such a long guide. So by doing this, we know that we can provide those guides in a cost-effective way to the community. And this is a mass spec. You use the mass spec to tell the ID of those long guides. So no matter it's HPLC grade long guide or it's a D-salt grade long guide, you are able to see the clean and the correct mass peak from the mass spectrometry we use. And aside from the mass spec, the question we got asked by the customer is, could you verify the sequence fidelity of the sequence? So as to answer that question, we have developed an in-house NGS sequencing method to verify the fidelity of those long guides. So here's an example. We can not only verify the correct sequence ratio, but also verify the improper sequence ratio. So to give you an understanding of what kind of impurity and what the ratio of impurity presented in our long guides. So last but not least, we have our cellular-based team is able to perform those editing experiments for us so we can better understand the quality-effective ratio. So in this case, we use the D-salt grade long guide RNA compared with an HPLC grade long guide RNA. You see that both guide RNA showed an increased editing efficiency with increasing of the dosage. As I just presented earlier, HPLC grade guide RNA has better purity compared to the D-salt. So relatively in each dosing amount, the editing efficiency was higher for HPLC grade. But the D-salt grade was comparable and is moderate. Using the maximum amount, it could reach 50%. It's a pretty good number for prime editing. GenScript has been the leading CRISPR nucleic acid providers, which can cover the discovery phase all the way down to clinical trials. We have adopted phase-appropriate QMS system, including the iOS 901, GMP-like, and CGMP quality system, so that we would allow you to help you coming from the discovery stage all the way down to the clinical trial stage. And GenScript has been provided clinical grade batch delivered through 2013, over 100 batches. And we have supported over 30 IND filing projects, and 6 of them have received IND approval across the CN, FDA, and EMA region. By partnering with GenScript, you get access to CRISPR guide RNA, RU to GMP grade. And not only this one, you also get access to DNA payload templates from RU to GMP, including the ssDNA, which show the lowest toxicity, and the double-strand DNA, which could allow us to do larger size insertions. We could also cover it from RU to GMP as well. And not only the materials, you would got the global filing support, which compliant with FDA, EMA, PMDA, and NMPA regulations. So I hope after this, I showed you that we could support you from discovery to the clinical trial in a quality-based way. If you want the maximum quality, we will provide you the HPLC grade material. If you want to get a taste of how prime editing works, we would also provide a cost-effective D-salt grade, which would work as well. So that's all for my part. Now I'm going to invite my colleague Fengmei to give the second one.
Yeah, thank you for joining our presentation. I'm Fengmei from Innovation Center. Our mainly cover about the mRNA platform and LNP delivery. So I'll introduce about our solutions and the technology advance for the CRISPR solution. As mentioned above, the guide RNA is one essential component for your success of CRISPR gene therapy. And the main challenge for the CRISPR delivery still resides in the delivery system. So traditionally, using genes as the vector, those rely on the AAV or virus-like particles for delivery. But with current advance of mRNA technology, using the combination of mRNA version to encode for the CRISPR enzyme and guide RNA, those can be encapsulated into LNP and delivered in vivo, with low risk of the high dose and low risk of the insertion mutation. So LNP is very familiar system that have been used in the COVID vaccine, which have those 4 essential components for effectively load RNA and mRNA to one vehicle for successful in vivo delivery. So at GenScript, we have delivered this system with optimized ESP-Cas9 mRNA and track sgRNA as a model. And we have been able to load them into the several different LNP formulations. By comparing this formulation with traditional lipofectomy as transfection tool, you can notice that with this formulation, you get a much higher gene editing efficiency in the HEC293 T cells here. And we also tested those systems in the GelCat cells and also primary T cells. They are all achieved a very good editing efficiency. So next, we have developed a more broader toolbox for you with different formulations and for different cell lines for effective gene editing. So here, these are the comparison data with the sLNP, aLNP, or LNP. Those are our LNP options that can have successfully achieve gene editing efficiency, gene knockout for the track guide RNA in both HEC293 T cells, GelCat cells, and the primary T cells. We're going to offer a total solution with this optimized LNP formulation service with tailored adjusted N2P ratio to ensure high encapsulation efficiency for both your mRNA and guide RNA. And also, we have titrated the ratio of mRNA and guide RNA with a good range for you to start with the project optimization. And also, how about the dosage? Those are also optimized. So for 0.2 million cells, we have found that with a dose of 0.6 microgram to 2.4 microgram worked pretty well. And we have also further improved this editing efficiency by improving the quality of the sgRNA, as mentioned before, and also mRNA. So mRNA reducing the immunotoxicity will help to improve editing efficiency greatly. Next, we also have been able to successfully using this system to deliver the knock-in solutions. So with gene knock-in, it will require 3 essential components, which are the single-strand DNA, where we have found that using the hybrid single-strand DNA HDR design has been greatly improved the lock-in efficiency, and also sgRNA and mRNA. So with this 3-in-1 LNP formulation, we observed great about 37% editing efficiency to knock in GIP into the HEC293 T cells. And those are also confirmed with sequencing and PCR experiment. So next, I also want to introduce, we have advanced tools that to further improve the mRNA quality to improve its expression efficiency. For those long mRNA for CRISPR, for Cas9 or Cas12a, the side effects, the side products of double-strand RNA is a problem to introduce immunotoxicity that reduces its expression efficiency. So we have been able to successfully optimize our IVT system and purification system to greatly reduce the double-strand RNA residue in the Cas9 mRNA, and then we observe a great improvement in the expression of the Cas9 mRNA after the optimization for mRNA system. So in overall, we are offering a one-stop solution from gene to mRNA and LNP. We can start from the gene synthesis, codon optimization, plasmid preparation, and mRNA IVT, and finally LNP formulation for your CRISPR editing solutions. So next, linear mRNA is a traditional solution that has been widely accepted for coding for Cas9 mRNA. We also have solutions with newer versions of RNA, which include the self-amplifying RNA and the circular RNA. So self-amplifying RNA uses the design of a replicase with RNA that can greatly increase its expression efficiency. And the circular RNA using a circular design with using the RES as a ribosome recruiting that help to greatly improve the RNA half-life in the cells and also expression efficiency. So current limit for these newer versions of self-amplifying RNA, the insert of gene of interest, you only limit to 6 kb. And for circular RNA, the insert limit is about 4 kb. And we have optimized the RES options for circular RNA that accommodates to different cell line expression. Last but not least, we also have those off-the-shelf mRNA and LNP products for gene editing solutions. Those are readily available in a rapid turnaround time in 5 business days. So those mRNA have like ESP-Cas9 mRNA, Cas9 circular RNA. We also have Cas12a mRNA, prime editing mRNA, and piggyback mRNA. And we also have a LNP that loaded with ESP-Cas9 mRNA and the track sgRNA as introduced in the earlier presentation data. So overall, at GenScript, we are offering a one-stop solution from screening to validation, scale-up, and clinical trials. We have the sgRNA HDR design tool, the sgRNA synthesis service, DNA payload template synthesis service, and the linear or circular mRNA synthesis LNP formulation, and also those off-the-shelf products for your platform proof of concept studies. Thank you very much for attending our presentation. Please visit us at our booth 807 if you have any more questions.